Before we continue talking about catabolic pathways, let's talk about how chemical reactions occur. All chemical reactions are based on oxidation-reduction reactions. Oxidation means removal of electrons. Reduction is gain of electrons. Let's look at this picture on this slide. When compound A reacts with compound B, as a result of this reaction, compound A will lose its electron, means it will be oxidized. When compound B will pick up this electron, it will be reduced. Keep in mind, only few electrons can exist in the free form, but usually when two compounds participate in chemical reaction, one compound lose certain number of electrons and the same number will be picked up by the second compound that participate in the same reaction. Because oxidation-reduction reactions are connected and balanced, we call them redox reactions. Let's repeat it. Oxidation is loss of electrons. Reduction is gain of electrons. In all redox reactions, one compound always loses electrons through oxidation. Another compound gains electrons through reduction. Let's look at this example of redox reaction from inorganic chemistry. So in this reaction, 4 molecules of iron react with 3 molecules or 6 atoms of oxygen. As a result of this reaction, 2 molecules of iron oxide will be formed. In this chemical reaction, 4 atoms of iron will react with 3 molecules of oxygen. As a result, 2 molecules of iron oxide are formed. So in this reaction, iron will lose its electrons and will be oxidized. Oxygen will gain those electrons and will be reduced. As I said, redox reactions are always balanced. And if you look at this reaction, we have 4 atoms of iron on the left side of reaction and the same number, 4 atoms of iron on the right side of reaction. We have 6 atoms of oxygen on the left side of reaction and the same number, 6 oxygen atoms on the right side of this reaction. This is an example of redox reaction in inorganic chemistry. Organic chemistry or biochemistry is a little bit different. In biochemistry, electron cannot be transferred from one compound to another compound in redox reaction. It must be attached to the proton. When you connect electron to proton, it will form hydrogen atom. It means in organic chemistry redox reactions, electrons will be transferred from one compound to another compound as a part of hydrogen atom. But please also remember, hydrogen atom also cannot be transferred from one compound to another compound in biochemistry. Hydrogen atoms require electron transport carriers, and those are NAD or NADP molecules. As I said, within redox reaction in biochemistry, hydrogen atoms are transferred from one substrate to another substrate or from one compound to another compound by electron transport carriers, and those are NAD or NADP molecules. And because electron carriers can accept hydrogen atom or give it away, they can exist in two forms or two states: reduced state and oxidized state. Reduced state looks like this: NADH, NADPH. You see hydrogen atoms are attached to electron carriers in reduced stage. And oxidized state looks like this: NAD plus, NADP plus. As you see, electron carrier in oxidized state loses hydrogen atoms. On this slide, you can see a chemical formula of electron carrier, and they show you that NAD or NADP molecule can exist in two forms. Oxidized form, NADP plus, means carrier lowest hydrogen atom, and reduced form, NADPH, you see here, NADP molecule gain hydrogen atom. And as you see, those reactions of reduction or hydrogenation and oxidation or dehydrogenation can go actually in both directions. Next, we will talk about ATP molecule or adenosine triphosphate molecule, which is a source of biological energy for any cell. Adenosine triphosphate molecule is composed of two main parts. It is adenosine, which is sugar, and three phosphate groups. Remember, the formula of phosphate group is PO3. PO3. Phosphate groups connected to adenosine with high energy bonds. High energy bonds are unstable. Every time when high energy bond is broken, energy will be released. But what is going to happen to ATP molecule when it loses its phosphate group? When ATP molecule loses its phosphate group, means gets dephosphorylated, it will become an ADP molecule, adenosine diphosphate, which is a low energy molecule. But the question is how cells can transform ADP molecule, which is a low energy molecule, into ATP molecule, which is high energy molecule. In order to convert ADP into ATP, cells have to attach phosphate group to ADP molecule. It means cells have to phosphorylate ADP molecule. One more picture of ATP molecule for you. This picture again shows you that adenosine triphosphate is composed of adenosine, which is sugar, and three phosphate groups. And those phosphate groups are attached to adenosine with the high energy bonds. On this slide, you can see if ADP molecule will go through reaction phosphorylation, then phosphate group will be attached to it. And of course, energy will be released in this reaction. As a result of this reaction, ATP molecule will be formed. But if we break one of the high energy bonds, energy will be released. ATP molecule will lose its phosphate group and will be converted again into ADP molecule, low energy molecule. This is an example for you of ADP phosphorylation reaction. The name of this reaction is a substrate level phosphorylation. Please remember the name of this reaction because cells actually use substrate level phosphorylation reaction to make ATP molecules. So on this slide you see some kind of substrate or nutrient is used by cell, and that substrate in its structure has phosphate group. When that substrate goes through phosphorylation reaction with ADP molecule, it gets dephosphorylated. It means substrate will lose this phosphate group. But ADP molecule will be phosphorylated because it will accept that phosphate group and will be converted into ATP high energy molecule. This is another example for you of a substrate level phosphorylation reaction. And here is some kind of substrate which has a phosphate group in its structure. It reacts with ADP molecule. As a result of this reaction, this substrate will be dephosphorylated because it loses its phosphate group, and ADP molecule will get phosphorylated. It will accept that phosphate group and will become ATP molecule. Alright, let's get back to catabolic pathways. Remember, in catabolism, there are three catabolic pathways: glycolysis, TCA cycle or Krebs cycle, and the pentose phosphate pathway. Altogether, those three pathways form 12 precursor metabolites. Let's look at this chart and talk about those pathways separately and a little bit more details. The first one is glycolysis. It forms 6 precursor metabolites. Also, glycolysis forms 4 ATP molecules by substrate level phosphorylation. 2 of those ATP molecules will be used in the beginning of glycolysis, means at the end of glycolysis there will be 2 ATPs left. What would be the end products of glycolysis? The end products of glycolysis are 2 molecules of pyruvate. TCA cycle or Krebs cycle forms 4 precursor metabolites. Also, it makes 1 ATP molecule, and the end product of TCA cycle is 3 molecules of CO2 or 3 molecules of carbon dioxide. And the last pathway is called pentose phosphate pathway. It forms the last 2 precursor metabolites. Please remember, it doesn't make any ATPs, it uses 1 ATP, and the end products of pentose phosphate pathway will be 3 CO2s or 3 molecules of carbon dioxide and 1 molecule of triose phosphate. Also, all 3 catabolic pathways produce reducing power compounds. Alright, let's repeat all information about catabolic pathways once again. The first pathway is glycolysis. It forms 6 precursor metabolites. It also makes 4 ATPs. Remember, 2 of those ATPs will be used in the beginning of glycolysis. So at the end of glycolysis will be 2 ATPs left. But what would be the substrate of glycolysis? The substrate of glycolysis is 6-carbon sugar glucose. What would be the end product of glycolysis? The end product of glycolysis is 2 molecules of pyruvate. On the next few slides, they show you glycolysis as a chain of biochemical reactions. For example, on this slide, they show you the first part of this reaction, and they show you that how glucose, which is 6-carbon sugar, gets split into 2 3-carbon sugars. And this is when those 2 ATP molecules are used. On this slide, glycolysis continue, and as a result, one more metabolite is formed. And also, please pay attention, the first 2 ATP molecules are made also here by substrate level phosphorylation reaction. And again, glycolysis continue, and as a result, one more, the last metabolite is formed, plus 2 more ATP molecules are made by substrate level phosphorylation, and as you see, the end product of glycolysis is 2 molecules of pyruvate. Next catabolic pathway is called TCA cycle or Krebs cycle. What you have to remember is Krebs cycle continuous glycolysis. The substrate for TCA or Krebs cycle is acetyl-CoA or acetyl coenzyme A. But if Krebs cycle continuous glycolysis, it's supposed to use as a substrate the end product of glycolysis, right? And the end product of glycolysis is pyruvate. So there's one more additional step here between glycolysis and Krebs cycle. It's called intermediate step. So intermediate step or transition step connects glycolysis to Krebs cycle because it converts one molecule of pyruvate, which is the end product of glycolysis, into one molecule of acetyl-CoA, which is a substrate for citric acid or Krebs cycle. Remember, please, transition step or intermediate step in eukaryotic or microorganisms will occur in mitochondrial matrix. Since prokaryotes or bacteria have no mitochondria, the intermediate step or transition step will occur in cytoplasm. So let's repeat again. The second catabolic pathway is called TCA cycle or tricarboxylic acid cycle or Krebs cycle. It forms 4 precursor metabolites. The substrate for Krebs cycle is acetyl-CoA, and in the end product it produces CO2 molecules, 3 CO2 molecules. Once again, this slide shows you that TCA cycle or Krebs cycle, it's a chain of biochemical reactions. It forms 4 precursor metabolites. 1 ATP molecule will be formed as a result of TCA cycle, and the end product of Krebs cycle or TCA cycle is 3 molecules of carbon dioxide. And the last catabolic pathway is called Pentose phosphate pathway. What you have to remember about this pathway is the substrate for pentose phosphate pathway is glucose again. And also you have to remember that pentose phosphate pathway, it is an alternative route between 2 points of glycolysis. So the question is why cells require this additional catabolic pathway. Because glycolysis and Krebs cycle or TCA cycle are not able to form last two precursor metabolites. Those are ribose 5-phosphate and erythrose 4-phosphate. Please remember the names of those last two precursor metabolites for your coming exam. So, as I said, the glucose again will be used as a substrate by pentose phosphate pathway. And then, as a result of chain of biochemical reactions, two metabolites will be formed, ribose 5-phosphate and erythrose 4-phosphate. No ATPs will be formed by pentose phosphate pathway, and the end product of pentose phosphate pathway is 3 molecules of carbon dioxide, 3 CO2s, and 1 molecule of triose phosphate.